Welcome to this explainer. Imagine, if you will, the human body as this incredibly bustling, highly coordinated megacity. Every single day, this metropolis is taking in huge shipments, right? Nutrients, sure, but also a massive array of foreign chemicals, medications, and byproducts. And look, just like any well-run city, if it's taking materials in, it desperately needs a world-class, highly efficient waste management facility to process and remove the trash. So today, we're going to explore exactly how your body's microscopic waste management departments identify, filter out, and permanently evict these chemical visitors.
Okay, let's dive into this roadmap for the explainer today. We'll be breaking this down into 5 distinct areas. First, the drug exit strategy. Second, the kidney's filtration plant. Third, secretion and cellular bouncers. Fourth, the liver's biliary route. And finally, other exits and variables.
All right, kicking things off with Section 1, the drug exit strategy, defining the big picture.
So, before we zoom in on the specific organs doing all the heavy lifting, we really need to understand the ultimate overriding goal of the body's waste system, which is, well, total elimination. And to grasp that, you've got to understand this crucial concept of clearance. Clearance is simply the volume of blood from which a drug is completely removed per unit of time, usually measured in milliliters per minute. Now, why does this specific metric matter so incredibly much? Because clearance dictates exactly what dose is needed for a drug to actually do its job. I mean, if your body clears a drug too quickly, it just won't work. But on the flip side, if clearance is too slow, that exact same dose is going to build up and become highly toxic in your blood. It is the body's ultimate biological balancing act.
And this brilliantly illustrates exactly what we're talking about. When we break down the routes of elimination for the top 200 drugs, you can immediately spot the heavy hitters. Notice how massive those top categories are? Metabolism and renal excretion are by far the dominant pathways here. Biliary excretion plays a smaller but still highly specific role. Today, our focus is squarely on those excretion pathways, how the body physically flushes the material out.
Moving right along to Section 2, the kidney's filtration plant, the master sieve.
The kidney essentially acts as the master sieve of the human body. Its absolutely crucial role is regulating our body fluids, controlling mineral composition, and meticulously flushing out those foreign chemicals we were just talking about. Looking at the macro-level anatomy, these organs are truly massive, high-volume filtration plants. Actually, despite being relatively small in physical size, the kidneys receive about 25% of our body's total blood supply at any given moment. 25%. That is a staggering amount of fluid continuously being routed into this organ for high-speed inspection and cleaning.
Zooming in a little further, we reach the real workhorse of the kidney, the nephron. This functional unit basically bridges the outer renal cortex and the inner medulla. And here's a truly fascinating fact for you. A single human kidney contains approximately 1 million of these microscopic, hard-working units. 1 million individual filtration lines operating perfectly in parallel. It's just wild.
All right, zooming in one last time to the very start of the nephron, we find a structure called the glomerulus. When we look closely at the blood vessels involved here, you've got a large incoming pipe, the afferent arteriole, and a much smaller outgoing pipe, the efferent arteriole. Now think about it. When you push a massive volume of blood through a large opening and try to force it out of a smaller one, you create a total pressure cooker environment. This resulting hydrostatic pressure literally forces fluid and molecules out of the blood and right into the kidney's tubules in a process we call ultrafiltration.
So, the crucial point is, to see this size-based sorting in action, let's look at the numbers. Up at the top, water. It's incredibly tiny with a molecular weight of 18, and it is 100% filtered. Glucose, slightly bigger at 180, also 100% filtered. But scan down to the bottom of the table. Serum albumin. This is a massive protein floating in your blood with a molecular weight of 69,000, and it has a filtration rate of less than 1%. So the glomerulus is essentially a physical barrier that simply blocks massive molecules while letting the small dissolved chemicals pass right on through.
Which brings us to Section 3, secretion and cellular bouncers, bypassing the sieve.
Now this size-based sorting poses an immediate, glaring problem for our biological city. What happens to the chemicals, toxins, or drugs that don't passively fall through the sieve? What if they're too big or tightly bound to those giant serum proteins? Well, that brings us to the difference between passive and active waste removal. Passive filtration is simply trapping chemicals in the kidney tubules due to their small size and the pH of the fluid. It just happens automatically. Active secretion, however, is a whole different ballgame. It requires the body to spend precious energy to physically move molecules across a cellular gradient. You can kind of conceptualize these active transporters as biological bouncers embedded right in the cell walls. They literally grab specific unwanted drugs from the blood, burn energy, and forcefully push them out into the urine.
Now, what's really interesting here is this fascinating mini-narrative of what happens when two different drugs encounter these cellular bouncers at the exact same time. Let's look at two drugs, penicillin, a very common antibiotic, and probenizid. They're both showing up at the exact same cellular exit door, trying to get out of the extracellular fluid and into the tubular fluid for elimination.
Let's break down the outcome of this cellular competition. Because both penicillin and probenizid are chemically similar, they're both weak acids, they get recognized by and have to compete for the exact same protein transporters. But probenizid is a bit of a bully here. It effectively blocks penicillin's access to the bouncer. Because probenizid is tying up all the exit doors, penicillin gets trapped in the bloodstream. The fascinating result? A significantly increased duration of action for the penicillin. In fact, medical professionals sometimes use this intentional traffic jam to make a dose of penicillin last longer in a patient's body.
All right, on to section 4, the liver's biliary route, the heavy lifters.
So while the kidneys handle that high-speed sorting, the liver steps in to handle the massive, bulky trash. If the kidneys are the fast-moving sieve, the liver is absolutely the heavy lifter. It manages the protein-bound waste that the kidneys simply aren't equipped to process. In this microscopic view of hepatocytes, or liver cells, we can see how they act as powerful chemical sponges. They can sequester excess chemicals directly out of the blood, and they have the unique capacity to physically extract drugs that are tightly bound to large proteins. Once they've extracted these large toxins, they move them into the bile canaliculi, where the waste is mixed into bile and sent down the digestive tract for ultimate removal in the feces.
But we definitely have to highlight the occupational hazards of this heavy lifting department. The liver isn't just moving chemicals around, it's actively trying to break them down through metabolism. And sometimes this biological processing backfires. The liver's attempt to break apart a complex drug can accidentally create highly reactive chemical species. These rogue metabolites can actually turn around and attack the liver itself, causing tissue damage known as hepatotoxicity. It is a dangerous, volatile job, but absolutely essential for our survival.
Finally, we reach section 5, other exits and variables, making it personal.
While the kidneys and liver undoubtedly do the heavy lifting for drug elimination, our bodies have actually developed several other, more subtle exit routes. Let's rapid fire through some of these alternative exits. Breast milk plays a specific role, easily trapping lipophilic, or fat-loving compounds and weak bases. Then we have exhaled air. I know it sounds a bit strange, but it's actually the primary exit route for volatile compounds, like the anesthetics used during surgery. And here's a surprising detail. Saliva is actively being researched right now as a totally new frontier for testing. Because drugs can secrete into saliva, scientists are looking at it as an easy, needle-free way to measure the concentration of active, unbound drugs floating in your system.
But I really want to emphasize the ultimate takeaway for you here. Clearance is absolutely not a one-size-fits-all number. Every single process we just talked about, the filtration rates, the cellular bouncers, the liver metabolism, it all fluctuates drastically based on your personal variables. Your species, your gender, your age, and your unique genetic background fundamentally rewrite the rules on exactly how fast or slow a drug clears your system.
Which leads me to conclude with this provocative question for you to think about. If your age, your gender, and your deeply embedded genetics quietly alter your chemical elimination rates every single day, just how incredibly unique is your personal biological filtration plant right now? The next time you take a simple painkiller or vitamin, take a moment to marvel at the customized million-unit waste management facility operating flawlessly inside of you. Thanks for joining me for this explainer, and keep staying curious about the amazing machine that is the human body.